Today we're going to finish off looking at the biochemical process of photosynthesis, right? So it's just last PowerPoint and this PowerPoint that we'll be looking at photosynthesis, and we're really going to go into photosynthesis a little bit more in detail today. It can get a bit challenging, but I really encourage you to follow along and actually draw diagrams like I'm going to show in this PowerPoint, because they really help to put the ideas together so you can understand that a little bit better. All right. Before we do that, let's have a look at these three quick questions. So pause the video here and have a go. Cool. So we're actually going to go through these in class. All right. So in the study design, we're still obviously in that photosynthesis dot point, right? But now we want to talk about the actual inputs, so what goes in, and the outputs, so what comes out of both the two phases, the light dependent and the light independent stages of photosynthesis. All right, light independent stage, we can also call it the Calvin cycle. It's just named after the guy that discovered it, Calvin. All right, and we're going to just talk about it in terms of C3 plants. You don't have to know what that means. All right, it just means that we don't have to look at it in terms of C4 plants and CAM plants, which are really complicated. So consider yourselves lucky. We don't have to know about the details of the biochemical pathway mechanisms, but I'm actually going to touch on this. I'm not going to go into extreme detail, but I'm just going to touch on it, because by having a little bit of an understanding of this, you're going to better understand the inputs and outputs. Okay. And then lastly, we have to talk about the rates, the factors that affect the rate of photosynthesis, including light, temperature, and carbon dioxide concentration, which we all looked at in year 11 biology. So last time we talked about the photosynthesis equation as this: 6 carbon dioxide molecules, 12 water molecules in the presence of sunlight and that pigment we call chlorophyll, becoming glucose, which is C6H12O6, which you have to really know what that is, 6 water molecules and 6 oxygen molecules. By the end of today's lesson, you should be able to turn that into this, okay? All of this is the same information, just slightly broken down into the two stages of photosynthesis: the light-dependent reactions, which happen in the grana. All right, remember the grana are those membranes that are green because it has the chlorophyll. Here's a great way to remember that the light-dependent reactions have to happen there because it's green, and the green is chlorophyll that's absorbing the light. So light-dependent, absorbing light, must be in the green grana. All right. The light-independent reactions, on the other hand, have to happen in the stroma, which is that cytoplasm-like fluid that's inside of the chloroplast. All right. As much as we call this light-independent reactions, they can't happen without the light-independent reactions. So it doesn't mean we don't. So it's not the dark reactions; it's light-independent reactions. Okay. Let's look at everything that we have here. 12 water molecules. 12 water molecules. All right. 6 oxygen coming out. So we had 12 water molecules coming in, 6 oxygen coming out, 6 oxygen coming out. Okay. 6 carbon dioxide coming in, 6 carbon dioxide coming in, 6 water molecules coming out, 6 water molecules coming out, and that very important product of photosynthesis, which is glucose. See, no much extra information. The only bit of extra information is actually these two guys, the NADPH2s and the ATPs. You don't have to know those numbers, 12 and 18, by the way. All right, but all you have to know is that this sunlight that's coming into the light-dependent reactions, we're getting energy out of that, and that energy is translated from these reactions to the light-independent reactions via these energy carrier coenzymes, which we have had a look at before in terms of enzymes. Cool? Let's break it down a little bit more. So first, the light-dependent stage only occurs in those thylakoid discs, which make up the grana, which are green, of the chloroplast, if there is light present, okay? And what we're happening is we're using light energy that's picked up by that pigment chlorophyll to actually break down 12 water molecules, so H2O molecules, and actually take out some chemical energy by that breakdown, okay? We take that chemical energy and we have to put it somewhere. So where we put that is we put it in the loaded forms of the coenzymes ADP and NADP+, all right? And what those loaded forms are is the ATP and the NADPH, okay? That is how we're going to carry that energy from the light dependent stage to the light independent stage. Let's watch a visualization of this. So here we have the electron acceptors. All right, these are the guys. Oh gosh, my video's gone wrong. There we go. The electron, the thylakoid. So this is a chloroplast. All right. So we have the NAD plus unloaded enzyme and the ADP unloaded coenzyme. So these are unloaded coenzymes. We have light coming and hitting that grana, right? It's going to break down the water, and that breakdown of water is going to provide energy in the form of electrons, forming the loaded forms NADPH and the loaded ATP. Cool? So what happens to the water molecules? Sorry. That water molecule actually takes in the energy from the sun. Right, as you can see here, right? By taking in the energy from the sun, we break the water molecule and we get hydrogen, all right? And that hydrogen is what loads the NADP plus into NADPH, all right? And we get oxygen releasing as a gas as a byproduct. But most importantly, we get ADP turning into ATP. ATP means energy, and we get to take that onto the next stage of the reaction. Okay. One little note is this NADPH molecule. All right. We're going to see a very similar molecule when we talk about cellular respiration. Very easy to know that this one's to do with photosynthesis. That P is going to stand for photosynthesis for us. So if you see NADPH, we know it's photosynthesis. If it doesn't have the P, it's going to be cellular respiration. Cool? All right. Let's go to the next stage. We've gathered some energy. We're going to go into the light independent stage, the Calvin cycle. So it doesn't require light, but it occurs in the stroma as long as the necessary inputs are available. Remember, we need that energy input as the coenzymes. So energy from the ATP and NADPH in the light dependent stage is used to actually take the carbon that we input in the form of carbon dioxide and we load it and turn it into glucose. Here's a simplified version of this, all right? We have the Calvin cycle, and we're going to have 6 carbon dioxide molecules coming into the Calvin cycle at different stages, and we're going to get ATP and NADPH providing the energy to break down those carbon dioxide molecules and make glucose. Let's look at this a little bit more detail. All right, so here we can see that we still have molecules of carbon dioxide undergoing what we call carbon fixation. All right, what happens in carbon fixation? We're going to provide some ATP, some NADPH, all right? And by providing the ATP, NADPH, we get these carbon dioxide molecules turning into C6H12O6. Do you have to know how that happens? No, you just have to know that we have an input of carbon dioxide and an output of glucose from this process, okay? All right, so let's summarize all of that. All right, so we had light coming into the light-dependent reactions, okay, and they're going to happen inside the granum, right? Those green thylakoid membranes, right? The chlorophyll is going to absorb the light energy, which is used to break down water molecules into oxygen molecules, all right? And that breakdown of water into oxygen is going to turn ADP and NADP into their loaded forms, which we call ATP and NADPH, okay? Those loaded forms, they simply serve the purpose of taking energy from the light-dependent reactions and taking it over to the light-independent reactions, which we also call the Calvin cycle, all right? So that energy is going to be provided to do that carbon fixing process, which means that we're going to take carbon dioxide, specifically 6 molecules of carbon dioxide, and through a series of process which we call carbon fixation, we turn it into glucose, which is for some reason written there in a funny way, but all this is really is glucose, okay? This process, by the way, unloads those coenzymes, so we have to actually take the NADP and the NADP back to the light-dependent reactions so they can get loaded back up. Cool? Alright, now that we've talked about photosynthesis and the inputs and outputs, we have to talk about the experimental value in photosynthesis, which we can see in terms of what we call the limiting factors to the rate of photosynthesis. Okay, photosynthesis is really affected by 3 environmental factors. The first one, which we're going to study in terms of our SAC, is the intensity of radiant light and the availability of correct wavelength. We'll go into this a bit further when we do our experiment next Friday. Okay? Ambient temperature, so different temperatures can affect this, and the concentration of carbon dioxide. Okay? These are the main 3 things that will affect photosynthesis. Under certain circumstances, one of these factors can become what we call limiting. All right, that is, it's in short supply and it's actually going to restrict the rate of photosynthesis increasing. Okay, so let's look at a bit of an example in terms of Miss French's strawberries. Okay, so Miss French was actually trying to grow some strawberries at the end of last year in a shady area next to her back fence, but for some reason they wouldn't grow at all. Okay, so Mr. Mallow, knowing everything about growing strawberries, he actually told Miss French that the plants actually need plenty of sunlight to grow and recommended that she move her strawberries to the middle of her backyard, where we're going to get more sunlight. Miss French moved those strawberries under a big tree where they had a little bit more sunlight, and this made the strawberries grow quicker. Mr. Mallow, though, he thought that we could still make those strawberries grow faster, so he cut down the big tree, giving the strawberries as much light as possible. But this didn't make the strawberries grow any quicker. Why? Hopefully you can work out what's happened is we must have a certain limiting factor here. All right? So, for example, when Miss French was trying to grow her strawberries in the shade, we had the rate of photosynthesis as it is in A here, all right, a low rate of photosynthesis because we didn't have enough light available for the maximum rate of photosynthesis to happen. But then Miss French moved her strawberries to the middle of her yard, which gave her a rate of photosynthesis that was optimum, all right? So it sort of looked like this, okay? It had a much higher rate of photosynthesis, more light was available. But if Mr. Mallow cut down the tree, this didn't actually increase the rate of photosynthesis because something else was limiting photosynthesis increasing. So this could have been not enough carbon dioxide, not enough water, all right? It could have been the temperature wasn't increasing or wasn't optimal anymore. Okay, so this could have been various things that could have limited the increase in photosynthesis. Carbon dioxide concentration works much the same. Okay, so as we increase the amount of carbon dioxide, so as we go up this slope here, we are going to increase the rate of photosynthesis. There's more carbon dioxide available to make glucose. Make sense? But we're going to get to a certain point where another factor has become limiting. Okay, so this in this example, it could be light intensity, for example. Okay, so much the same. Temperature, on the other hand, we're going to get something that looks a lot like the temperature enzyme reactions. Okay, where as we increase the temperature, photosynthesis will increase until it reaches what we call the optimal rate for photosynthesis. Okay, and it's happy there. Photosynthesis happens really fast. But if we decide to increase the temperature a bit more, what's going to happen? Enzymes denature, okay? All right? And this is pretty obvious, right? If you put a plant in an oven, it's not going to grow. It's going to die, right? But if you have a plant in a nice, warm, sunny environment with all the things that it needs, it's going to grow really well. If you put a plant frozen outside, if it's 0 degrees, it's obviously not going to grow. So this is actually pretty straightforward, isn't it?